By the late 2020s, humanity will generate roughly 180 zettabytes of digital information every single year. We have mastered the art of data creation. The challenge now is data retention. Storing even a fraction of this output requires a massive footprint of real estate, thousands of miles of copper wiring, and power-hungry cooling systems. Traditional digital storage represents a strict physical compromise. Every hard disk drive or solid-state memory bank relies on finite physical space to house magnetic domains and transistor gates. We are running out of that space. Manufacturers are rapidly approaching the absolute atomic boundaries of how small a single bit can be etched onto a silicon wafer. Meanwhile, the exponential surge in AI training sets, high-definition video, and genomic data is outpacing our capacity to physically build new warehouses to hold it. Continuing to scale up our existing silicon infrastructure is no longer a viable mathematical solution. The data volume is forcing a reevaluation of what we use as a storage substrate. The burden extends beyond physical real estate. Maintaining idle data requires enormous energy consumption. The constant rotation of spinning platters makes data centers a significant, growing drain on the global electricity grid. Physical media also rapidly decays. Even the most robust magnetic tape used for archival storage chemically degrades within a narrow 10 to 30-year window. Mechanical hard drives are far more fragile. Under continuous enterprise use, their complex physical mechanisms typically fail within 3 to 5 years. To prevent bit rot, enterprises are forced into a constant cycle of migration, actively copying their massive datasets to entirely new physical media. This continuous migration is a never-ending relay race against format obsolescence, costing the tech industry billions of dollars just to keep existing archives alive. Because of these unavoidable physical trade-offs, synthetic biologists are proposing a radical pivot. Rather than developing another silicone-based medium, they are borrowing a storage system perfected across 3.7 billion years of evolution. Deoxyribonucleic acid is highly stable, dense, and acts as a battle-tested data storage polymer. At the hardware level, every digital file, whether it is a JPEG, a PDF document, or a complete executable operating system, reduces down to a long string of binary ones and zeros. DNA encodes the operational blueprint of a living organism using a similar architectural logic. But instead of a base 2 binary system, it utilizes a base 4 alphabet consisting of adenine, cytosine, guanine, and thymine. This chart shows the central mapping table engineers use to bridge these systems. Because grouping binary into pairs of bits yields exactly 4 unique values, those pairs perfectly match the 4 available nucleotide bases. The assignment is direct. A 0-0 binary pair maps to adenine, 0-1 maps to cytosine, 1-0 to guanine, and 1-1 to thymine. When applied to an 8-bit digital snippet like 00011011, the binary decomposes into the genetic sequence ACGT. By utilizing this mathematical mapping, absolutely any digital payload can be algorithmically converted into biological syntax. A raw translation is not sufficient on its own. The physical processes of biological synthesis and sequencing frequently result in insertion, deletion, and substitution errors. Biology also imposes specific constraints. Long repeating runs of a single base, known as homopolymers, are difficult for sequencing machines to process without losing their place. To stabilize the sequence, data scientists inject redundancy algorithms directly into the biological code. Using methods similar to the Reed-Solomon codes that protect CDs, they break up these unstable repetitive chains. Massive digital payloads must then undergo fragmentation, breaking the continuous data stream down into thousands of short, biologically manageable synthetic strands. Because the file is now shattered, each individual fragment is tagged with a unique genetic address sequence, ensuring the data can be reassembled later, functioning exactly like a digital file path. This process lifts the error correction architecture of pure computer science and applies it directly to organic chemistry. Once the biological code is finalized and error-checked, the sequence data is exported to a DNA synthesizer. Through a process called phosphoramidite chemistry, the machine physically constructs custom strands base by base, linking each nucleotide together in the exact order specified by the file. This synthesis makes the digital file a tangible physical molecule. For long-term storage, this synthesized DNA is dehydrated. It sits inert in a small glass vial at room temperature. When the data needs to be retrieved, the DNA is rehydrated and run through a standard genomic sequencer. The sequencer outputs the biological bases, and a reverse mapping algorithm reconstructs the exact binary output. This creates a lossless, closed-loop cycle, translating digital code into physical matter and back into digital code. When measured against standard silicon enterprise storage, the physical density of this medium is unparalleled. A single gram of DNA can theoretically hold 215 petabytes of data. That is the equivalent of hundreds of millions of gigabytes contained within a microscopic footprint. Its longevity is equally significant. Under cool, dry conditions, DNA remains stable. Scientists have successfully sequenced intact genetic instructions from Ice Age permafrost specimens that are tens of thousands of years old. When compared to the fragile, decade-long functional lifespan of enterprise hard drives, biological storage extends archival integrity by millennia. Furthermore, biological storage achieves high energy efficiency. Once synthesized, the DNA requires zero continuous power or cooling systems to preserve the data. These three metrics, density, longevity, and efficiency, make DNA a specialized candidate for long-term cold storage that current enterprise systems cannot match. There is, however, a current engineering bottleneck. The chemical synthesis required to write DNA data is significantly slower and more expensive than writing to standard flash memory. There are also limitations on random access. You cannot instantly jump to a specific file within a pool of DNA. Retrieval requires amplifying and sequencing massive batches of the biological material, and then using computational filters to locate the exact fragment. Because of this friction, DNA is positioned strictly for vital, rarely accessed archival data, such as legal records, cultural heritage, and scientific datasets. It is not replacing local RAM. But for archives, DNA addresses a persistent problem in data retention. It eliminates the risk of format obsolescence. A hard drive built today may be unreadable by future hardware, but as long as biological life exists, the imperative and the technological capability to sequence DNA will be maintained by human civilization. By reframing data compression as a material science reality, we are learning to write our digital civilization directly into the molecule of life.